everyone, and welcome to today's webinar focused on the new Whole Slide Triage Imaging application. We have two speakers today. First, we have Jeff Hartford, who is the Senior Product Manager here at Lycor Biosciences. Dr. Kimberly Samco will also be joining us. She is the Principal Investigator at the Samco Laboratory in the Center for Imaging Medicine in the Thayer School of Engineering at Dartmouth College. During this webinar, we will be having live panelists who can answer any questions that you may have. Please feel free to submit questions at any time. Now I will pass it off to Jeff.
Well, thank you, Lauren. Before Dr. Samco shares her research, I wanted to briefly set up a new paradigm that I'm calling Whole Slide Triage Imaging. Now, the premise behind Whole Slide Triage Imaging is simply to make slide imaging more efficient for researchers. Now, although slide imaging does take place in both clinical and research labs, I'm going to be focusing my discussion on slide imaging that's only in the research lab. So slide imaging is a fundamental application that's in the research lab. Tissue sections are commonly stained with H&E, and then for colocalization studies, sequential fluorescence sections will also be imaged and then overlaid with the H&E slide. This process, depending upon how you look at it, can be a costly endeavor. It requires specialized equipment, it's time-consuming, and it requires a sophisticated storage infrastructure for the resulting data. And given these current challenges, the concept of whole slide triage imaging offers a potential solution to improve the current technologies. Now, to appreciate this, let's take a look at the progression of slide imaging. Traditionally, pathological samples were put onto slides which would then be taken to a microscope in order to be visualized. When the relevant region of interest was found, a picture was able to be taken of that region. However, there was anticipated value in having the ability to digitize the entire slide, not just a small region of interest. And there was also a need to somehow expedite the acquisition and visualization process of these slides. This led to the concept of whole slide imaging. With whole slide imaging, a specialized imaging system was able to digitize an entire slide at high resolution, which now allowed the full slide to be visualized and analyzed at a separate computer. Now, this was a huge advancement for digital pathology research, as the entire slide could be observed both remotely and through time. In addition, it was much faster than the traditional approach. That said, whole slide imaging is not without its limitations. Acquired images generally are high resolution and large, and normally they exceed about a gigabyte or more per channel for a 15 by 15 millimeter section. Further, whole slide imaging systems are expensive. And although they improve on the efficiency over traditional microscopy methods, the image acquisition can still be time-consuming. These systems are also limited in their ability to assess thicker tissue sections, which translates into the need for imaging more slides. And finally, with the increased demand for multiplexing, whole slide imaging systems generally are not set up for optimal detection in the infrared wavelengths. Now, when you consider the number of slides that need to be imaged in a lab, it seems apparent that storage challenges and time consumption are significant burdens to a lab. And this is where whole slide triage imaging comes in. The idea behind whole slide triage imaging is to rapidly screen all slides at a lower resolution and only move the relevant slides forward for microscopic imaging. With whole slide triage imaging, you can quickly capture the H&E and the fluorescence slides at the same resolution. The resolution of 5 micron is high enough to provide screening value, but low enough to keep the size small. Since the images are the same resolution and a slide holder keeps each slide perfectly aligned, the multiplexing images are easily overlaid for assessment on which slides need to undergo further microscopic analysis. And finally, thick tissue sections may also be imaged, which means that you may not need as many slides to screen. So what kind of time efficiency can be realized with whole slide triage imaging? Well, if you compare the Odyssey M against a typical whole slide imaging system, you can realize about 75% time savings. And when you begin looking at imaging large numbers of slides, this translates into a lot of time that's saved. For example, just imaging 25 pairs of H&E and fluorescent slides could take well over 4 hours on a traditional whole slide imager. But the same number of slides could be imaged in just over an hour on the Odyssey M. In addition, looking at the storage efficiency of whole slide triage imaging, the acquisition of those 25 pairs of slides will result in just under 2 gigabytes of data on an Odyssey M. Compare that to over 150 gigabytes on a traditional whole slide imaging system. This truly demonstrates the value of screening slides and only moving the relevant slides forward for the subcellular imaging. All of this is possible with the Odyssey M Imaging System, but given its extreme versatility with 18 channels of detection, the system can also be used to do membrane assays, gel assays, and plate assays, in addition to the tissue imaging. So given that background, I would now like to turn the webinar over to Dr. Kimberly Samco for you to understand a little bit about the work that they're doing with the Odyssey M in their research.
Hello, my name is Kimberly Samco, and I'm Associate Professor at the Thayer School of Engineering at Dartmouth College. Today I'm going to tell you a little bit about how our lab uses the Odyssey M whole slide imaging for fluorescence-guided surgery. Our research really focuses on molecular imaging, and that even further focuses down on fluorescence. You can see by this graph on the left-hand side that optical imaging has a wide spatial resolution from sub-micron all the way up into millimeter resolutions, and yet still maintains really high molecular sensitivities in the nanomolar range. These two resolutions combined far supersedes any other clinical medical imaging technology. For instance, PET has high molecular sensitivity but low spatial resolution, and MRI has low molecular sensitivity but high spatial resolution. To illustrate this even further, as you can see on the left-hand side or the right-hand side, is fluorescent images of tumor tissue in a patient at the top, and excised as a whole gross specimen, and then as bread loaf slices as we move down. As you can see, we're able to observe the fluorescence in the in vivo situation at low spatial resolution, and we can increase that spatial resolution as we go and the tissue travels through the pathology. In fact, we can even start to see cellular patterning down at the submicron range, and we could also take a microscope and look at the cells at the actual expression level on the cells themselves. The clinical agent that we use most consistently is ABY029, which is an anti-epidermal growth factor receptor alpha body molecule that was designed in collaboration with Alpha Body AB and Lycor Biosciences. We have taken this agent through 3 clinical trials in glioma, head and neck, and soft tissue sarcoma. The reason that we like this agent is that we have high EGFR specificity, no adverse events in humans, and also low molecular weight, such that we can inject the agent and have suitable tissue contrast in a matter of hours. However, after completing 3 clinical trials with this agent alone, we have come to the realization that we still have a significant amount of nonspecific fluorescence coming from the surrounding normal tissue. To explain this a little bit further, I have these diagrams here. If we think of a targeted agent like ABY029, we can observe it it is injected into the bloodstream, and it extravasates into the tissue, normal or tumor, and it can bind to a receptor of interest if the receptor is present. As you can see here by these concentration curves, the solid line shows the uptake of ABY in a tumor over time, and comparatively in that of normal muscle, which is devoid of receptor. You can see that these differences are quite significant. However, we see similar trends even with non-targeted agents. You can see here for a non-targeted agent that is incapable of binding to any receptor, we still see significantly higher uptake in a tumor, and this is due to the inherent property of tumors to have enhanced permeability and low removal, so there's higher retention within the tumor. So what we've done here is we have, so in the fluorescence images, it's hard to know whether the signal is coming from the tumor directly, from the agent actually bound to the receptor of interest, or if it's just in the tumor because of this EPR effect, or if it's just still nonspecific uptake in the surrounding tissues. So what we've done is we've come up with a computational model that uses a non-targeted dye that's injected simultaneously with our targeted dye, and we're able to remove the contribution from the plasma and the free space, so anything that is not bound to a receptor, and we're left with only the signal that's coming from the bound receptors. We can produce a secondary image, which we call a binding potential map, which is simply a ratio of the non-targeted or the targeted to the non-targeted image. And as you can see here, our targeted image is taken in the 800 channel of the Odyssey as IR dye 800 CW, and our non-targeted agent is typically IR dye 680 LT. We can produce this map, which is actually a map of receptor concentration and not a map of fluorescence intensity. We are using this application to try and improve fluorescence-guided surgery, specifically in the oral cavity of head and neck cancers in patients with head and neck cancer. Over 1 million people per year are diagnosed with head and neck cancer, and 10,000 deaths per year are anticipated. The goal of surgical treatment is to completely remove the tumor with clear margins, which means that we're not leaving any of the margins, any tumor behind in the surgical bed. However, in oral cancers, there is a true balance between taking the tumor out and preserving healthy normal tissue, which is responsible for functions such as eating and speaking and drinking. Unfortunately, with head and neck cancer, most of the decisions of whether the tumor was completely removed or not are made post-operatively, in which only less than 1% of the total tissue that was taken out is analyzed. Therefore, there are high levels of false negatives. It is virtually impossible to test all of the tissue using traditional pathology techniques because there is just too much and it's too time intensive. Therefore, we propose to use our fluorescence-guided technology, paired agent imaging, in the operating room and in the pathology suite to improve fluorescence-guided surgery. The general plan would be that after tumor or tissue was excised, the tissue is sent to the pathology suite in which it is sectioned, either frozen section, which takes about 30 minutes, or it can be permanently fixed in formalin and paraffin embedded, which takes 24 to 48 hours, usually for the analysis. After the tissue is sectioned and stained with H&E stain, a pathologist will read the slide and say whether or not there is any residual tissue in the tumor margin. What we propose is actually using paired agent imaging at the tissue level and at the section level to determine whether or not there are regions that are suspicious to have positive tissue. As you can see here in the bottom, this is an example of a tumor a tumor in a mouse tongue. Our previous workflow for doing this in a preclinical situation was using the Odyssey CLX. We collected our fluorescence images on the Odyssey CLX in both the 700 and the 800 channels, typically at 42 micron resolution, which takes several minutes to scan. We then had complementary images on consecutive tissue slices taken and scanned on the Leica Aperio, or the Perkin-Elmer-Vectra, in order to obtain digitized images of the pathology, the H&E and EGFR immunohistochemistry. We then, just to state, the Apereo is 20 times magnification, so high magnification and a line scanner, and the Perkin-Elmer-Vectra is actually needs to be stitched tiles. And so these are both very time-consuming, and they're also very, very large in size. Typically, each image is about a gigabyte, if not larger. In addition to these being large files, they're also very high resolution, higher than what we need in order to compare to our fluorescence. Therefore, we have to crop and resize all of the images in order to match the resolution of the fluorescence. And then we had a time-intensive alignment that we had to perform, in which we did both rigid rotations and nonlinear transformations. And nonlinear transformations is really just sort of stretching the tissue out in regions or shrinking the tissue in regions in order to match more closely. As you can see indicated here on the right, these dots were actually placed by hand by the person performing the analysis on both the pathology and the fluorescence images. And then we had to perform these alignments using MATLAB. So this entire process is very time-consuming. However, it gave us some really nice data that allowed us to directly compare our fluorescence data and our binding potential maps from our paired agent imaging to pathological images to show that we get significantly higher diagnostic detection. We have since modified this workflow using the Odyssey M and also gaining additional knowledge in the process. But for image processing, we now can produce both our fluorescence images and our RGB images on the Odyssey M using the fluorescence channels 700 and 800 channels and the RGB true color scanning feature. We can also scan all of these images at the same resolution, and if they are tissue sections, we use the slide scanning system that comes with the Odyssey M and allows us to repeatedly or reproducibly image slides. We've imaged down to 4 micron thick slides. So here's an example of the fluorescent images taken. These are 4 micron thick tissue sections. And then the nice thing about this as well is we image these before they're stained. We send the tissues back to the pathology lab. They stain them with H&E for us. We scan them on the Odyssey M in RGB true color, and we send them back to the pathology lab where they're destained and then restained with EGFR immunohistochemistry. And therefore, we actually can get all of the signal that we need for analysis off of one tissue sample, and we can scan it on one system at the same resolution. And the only real adjustment that we need to make is some rigid rotation or rigid translation in order to align the images, because often the ROIs are not in exactly the same place between fluorescence and RGB imaging. But this is really minor, and there's actually programs in MATLAB that do this automatically for us. We can then produce very detailed maps of regions of true positive and true negative regions for diagnostic determination. And as you can see here, these results show very nicely that our binding potential map has the most area that is true positive and the most area which is true negative. And it's not surprising, but a lot of the uncertainty comes in the margins. And this is where we will move forward with computer learning or large data set comparison in order to really determine if we can improve the diagnostic accuracy. In addition to oral cancers, we are trying to move this imaging technique into Mohs surgery. Mohs surgery is a surgery that is performed on small skin cancers, in which a process called en-face margin analysis is used. In skin cancers, they are on a surface and they tend to be small. So what happens is the surgeon will remove the visible tissue, but there tends to be sort of these tendrils that go down deeper into the surface that we cannot see by eye. So what the surgeon does is he removes small amounts of margin and then sections those and then performs H&E in order to diagnose whether there's any tissue remaining. As you remember from the previous slide, frozen sections can take 30 to 40 minutes in order for them to be frozen, sectioned, stained, and then looked at by the pathologist. In the meantime, the patient remains in the surgery room or in a waiting room for the results, If the results are positive, as you can see here in sections 1, 3, and 4, the surgeon will go back and remove more tissue in those regions and repeat the process. As you can see in this example, we still have positive staining in sections 1 and 4, and so additional tumor sections are removed until we get negative margins in all regions. Now, this is a very time-intensive process, taking several hours per patient, especially if more than one additional margin needs to be taken. In addition, the more margins that we have, and the larger the tumor, and the more times the surgeon has to go in and take another margin, means that we have an additional number of slides that needs to be read. So in a typical tumor that is 1 to 4 millimeters in diameter, you can read up to 30 slides per patient. But in any tumors even larger than that, it actually increases exponentially with the surface area that you have to analyze. And so you can see that up to a tumor that is 16 millimeters in diameter, you would have to read almost over 450 slides, which really makes this process only viable for small tissue, small tumors such as skin cancers. So we're working with a team of surgeons and pathologists to bring paired agent imaging into this process in order to try and identify suspicious regions without having to perform as many diagnostic pathology sections. Here you can see an experimental setup where we have tried to mimic the en face Mohs surgery in a mouse. We have subcutaneous tumors where we remove the bulk tumor as they do in the surgery, and then we remove 2 margins, one from the bulk tumor that was removed and one from the deep margin. We can image these on the Pearl Impulse, and what you can see here is that the fluorescence signal is bright in the bulk specimen, which we anticipate, but we actually do see some signal coming from the deep margin as well, which would indicate that there was some tumor left over. The nice thing about the Pearl is that we actually get a little bit more depth sensitivity due to the wide field imaging. However, these same tissue samples were scanned on the Odyssey M, and we can get very high resolution detailed images of the fluorescence on the surface, indicating whether or not there are actually any tumor cells on the surface or very near to the surface, which is what the surgeons are concerned about. Furthermore, we can take, to clarify, these images on the Odyssey M were taken on the flat glass surface using a custom scan. However, what we do is we pathologically section these serially, and we take sections every 200 microns approximately, and we again scan the frozen sections or the paraffin embedded sections for fluorescence, and then we can correlate those to the pathological images. So you can see now that we can actually rapidly scan a high number of sections and get very large correlating data sets, which is really important for trying to prove the diagnostic ability of our technique. So one of the things that we're able to do from these slide scans is we can look at the distribution of our signal in the images. Here you can see we're comparing paired agent imaging to single agent, which is that ABY029. As you can see, the signal is fairly bright all over the entire region for both. But if we actually just look at the signal that comes from one standard deviation away from the mean of the image, we can see that we are seeing these really hot spots here. Now, there's a theory that these hot spots are going to correlate to sentinel margins, so positive margins. And what we found in our tissue samples is that the paired agent imaging actually paired really well, matched really closely to actual regions that had tumor in them, whereas the paired agent imaging did not. As you can see here, we have pinpointed that in the first 600 microns, so the first 3 sections from the surface, we can identify the closest region to the surface was region 1, which is the brightest in our paired agent imaging. Region 2 comes in 200 microns deeper, which was the second brightest, and then we have section 3 over here in the corner, which came in at 600 microns, and you can see that this is correlating to these 3 hotspots, which could be considered sentinel margins. So we are very excited about this data, and we believe that paired agent imaging correlates well with our pathology and provides both highly correlated spatial information as well as depth information. We are currently bringing paired agent imaging into the clinic, and will hopefully be collecting some human data within the next year or so. So overall, I would like to just summarize in saying that Odyssey M streamlines our whole serial slide imaging for fluorescence-guided surgery applications. It has very rapid scan times using the line scanning feature of the Odyssey M compared to a raster scanning of the Odyssey CLX, and other imaging techniques that have much higher but unneeded resolution. We are able to collect both fluorescence and RGB true color, which are hardly spatially correlated, and eases in our ability to collect information. It also results in much smaller file sizes because we're collecting so many images, both of different imaging types or different staining types plus serial sections. In a matter of just a few mice, we can collect hundreds of tissue sections. And so this really streamlines our system and also allows us to have manageable data files. So I would like to just acknowledge the people from my research group who did this work. Chung Wang, who recently graduated with a PhD, has done all of the work on the oral cavity fluorescence-guided surgery, and Veronica Torres, a postdoc in the lab, is working on the Mohs surgery. In addition, I'd like to thank my collaborators from both Dartmouth College and Illinois Institute of Technology. And of course, this work was funded by the NCI in collaboration with an industrial academic partnership with both LICOR and AlphaBody AB. Thank you for your attention.
Thank you, Dr. Samco, for that great presentation. We've gotten a couple of questions in from the audience for you. The first one is, can the Perkin-Elmer instruments also detect the 800 nanometer dyes?
I can speak about the Perkin-Elmer systems. We have several at our institute, a variety of different types of IVUS systems and Maestro systems. Both of these are capable of imaging in the near infrared. If you buy the correct version, they have both visible and near infrared versions. However, these are more of in vivo imaging systems and aren't good for imaging slides. However, we also have a Vectra 2. The Vectra is a slide scanner. It's capable of imaging 6 slides at a time, and it comes in both a visible and a visible plus near infrared version. Our institute only has the visible version, so we are not able to image the 800 nanometer dyes on the system. However, I do know that they are available. One of the big differences I find between the Vectra 2 and the Odyssey M system is that the Odyssey M is made for whole slide scanning, where the Vectra 2 really isn't. It's made as a system that does high resolution tiles. And so they actually do like a whole slide scan as a pre-scan basically, but really that is not the intent, and it's actually difficult to export as well. Really only through a series of screenshots can you get to the whole slide scanner. So the Vectra is actually good for subcellular regions, where you're looking at fine cellular detail up to, I think, at least 40 times magnification. Whereas the Odyssey M is better for whole slide scans, where you're really looking at several millimeters or even centimeters of tissue at the same time.
All right, thanks for that answer. The second question we have is, can the Odyssey detect subcellular structures such as the nucleus at 5 microns?
The Odyssey M is not capable of imaging subcellular structures at 5 micron resolution. Really only macroscopic details of the tissue structure are visible, and this is really a feature for whole slide scanning rather than microscopic subcellular structures that you would achieve in a high resolution microscope.
All right, great. Our third question is, on the 200 micron sections, is the Odyssey M able to detect signal if it is deeper in the tissue rather than on the surface?
We have noted that the Odyssey M is able to detect signal deeper in the tissue rather than a surface for thicker sections. So when we use our fresh tissue where we have thicker tissue sections, we are able to image at a greater depth. So this actually scales with resolution. So actually at high resolution, you favor a more surface weighted. And so at the 5 and 10 micron settings, it's going to appear more similar to very thin cut sections. Whereas if you decrease the resolution, you actually increase the depth penetration of the light, and you actually can get some deeper depth penetration. Just note that this is for fresh tissue. We cut our tissue sections for pathology at between 4 and 7 microns in thickness. And we really don't notice a difference depending on resolution for these slices because they are so thin. We really only notice a depth penetration difference in the fluorescence images with our fresh tissue sections.
Our next question is, do you find that the 5 micron resolution of the Odyssey M is adequate for most of your presentation and publication needs with tissue section slides?
Yes, we definitely find that the 5 micron resolution of the Odyssey M is more than adequate for most of our presentation and publication needs. In addition, a lot of our image analysis is done off of these images as well. We find that presenting whole slide images, you really don't need the resolution that can come from slide scanners, especially those that are used in the pathology department that image at 20 or 40 times magnification. Those are nice for looking at subcellular regions, and so if you really wanted microscopic images of your slides, then you would want to choose a different system. But the 5 micron resolution on the Odyssey M gives really nice, crisp images that are suitable for presentations and for publication. You've seen some of them in my presentation today.
All right, we're to our last question here that we're going to ask Dr. Samco on this webinar. If you have more questions, please continue to submit those and we'll get those to our panelists to answer. The last question is, how much has it increased your productivity and efficiency to have a system like the Odyssey M that is compatible with so many different imaging needs and requirements?
The Odyssey M has really been beneficial in our lab for increased productivity. One is in our fluorescence studies, we no longer have to work really hard at aligning our fluorescence images with other systems, which often require changes in resolution and then both and rigid transformations. Most of the time when we're doing our alignment now, it's just a very simple, rigid transformation just to make sure that the fluorescent and the pathological images are aligned correctly. However, in saying that as well, compared to slide scanning on, let's say, the Vectra we could do 6 slides, but because it was scanning at such high resolution and in tile format, it actually took an hour or more to do 6 slides. So this was very time intensive and labor intensive on the part of the worker. In comparison, right now, we are undergoing a study in which we are imaging serial sections through entire tissue samples. And so in this case, we have a current situation we were discussing last week, was that we had 4 tissue samples, which accumulated over 500 slides. In this case, we are allowed to do 6 to 8 minutes for 12 slides. And so although this is still time consuming on our part, it greatly reduces the amount of time that we're spending compared to the Vectra. So just to sort of reiterate that would be 12 slides over a 6 to 8 minute period versus 6 slides over an hour. So this is really a much faster way, but again, with just the realization that this is more of a publication quality or presentation quality image versus a microscopic image. But for us, that is approximately 90% of our needs. If we need microscopic, then we have other instruments available at our institution, and we can selectively image those slides instead of imaging all of them like we do on the Odyssey M. It's really so convenient to do the whole slides.
Thank you, Dr. Samco, for presenting your research and answering those questions. Also, thank you all for attending our webinar. We hope that you've learned something new about tissue section imaging and how whole slide triage imaging can save you valuable time and resources. If you have any questions about today's webinar, please feel free to reach out to us at biosales@lycor.com.